So we're going to continue on with the cardiovascular system and heart. And I just want to bring your attention to, there's some really interesting, I call it hot heart research, cardiovascular related research, and regenerative studies looking at the heart. So actually we can use stem cells to study the human heart, its development, its growth, and its function. There's a lot of promise, future promise for regenerative medicine. So in doing studies where cardiovascular disease is attempted to be repaired, or heart damage after a heart attack, a myocardial infarction, or an MI, using stem cells to try to repair the damaged tissue. There's already clinical trials using stem cells in heart studies all over the world. There's some in the US and there's a lot in many other like Latin American countries and Asian countries. They're already doing stem cell work and looking at specifically at the heart. So I want to show you, this is a really interesting, so you can see that we can take stem cells. A lot of the clinical studies, that means focused on people and looking for potential regenerative medicine applications. A lot of those studies, they will use straight stem cells, either embryonic stem cells from a cell line that's been grown in the dish, often many for a long time, or they'll use induced pluripotent stem cells. That means taking a skin sample, turning those skin cells, those fibroblasts, into stem cells, and then putting those in the tissue to try to repair or fix damage. They've already done a lot of studies like this in rodent models and in monkeys and in lots of different animals, and now those are starting to translate to the human. And I already talked about, right, in the eye with rods and cones and growing stem cells and putting them into that tissue to help regenerate those damaged neurons. This is showing that we can take stem cells in the dish that came from someone's skin. So you get a core little biopsy punch, they do it up onto the inner aspect of the arm, and then plate those down in the dish. And over time they're going to grow, and the fibroblasts, the ones that secrete the fibers of skin, remember we talked about fibroblasts, and they make the elastin and reticular fibers and collagen. The fibroblasts actually, if we treat them in a specific way with a certain cocktail of factors, transcription factors that change gene expression, pretty much people worked it out to these 4 key factors that are needed. If you treat those fibroblasts from a skin sample that way, they actually de-differentiate. So I don't know if you get what that means. If a cell differentiates from the embryo, it specifies and gives you unique anatomy and morphology and physiology, right? We talked about all the cell types and tissues. That means going backwards, sort of pushing it back up the hill and de-differentiating it, making it more like an embryo-like cell or an embryonic-like cell. This has revolutionized physiology, medicine, biology. The ability to de-differentiate, or we call it, basically it's cellular reprogramming. You are reprogramming a cell to take on a new identity. And this really started, there was first publications in 2007, and now just tons of them since then. Actually, the scientists that won the Nobel Prize in Physiology or Medicine, it was for cellular reprogramming, cloning, and creating iPS cells. What are iPS cells? Induced pluripotent, remember this? Stem cells. Or people in the field will just call them iPS cells. That means we can get a skin sample, or we can get any other kind of fat. We can take adipose cells, we can get fibroblast cells, we can get blood cells, all kinds of different components, and actually de-differentiate them and turn them into these stem cells. So now the cool thing about these is they don't come from an embryo. So the original development of creating that line didn't involve an embryo. They're typically from an adult, okay? Now they can be used for a lot of these stem cell studies. And you can treat them, they become an immortalized line, they can divide and keep dividing. They're pluripotent. What is that one key word, pluripotent? What does that word mean? Pluripotent.
So like they're capable of differentiation?
Yes, that's right, Tony. They're capable of differentiation. That means now they can turn into a lot of different cell types. I can harness that. I can give them a certain series of factors in the media, and they can turn into specific cells. So it is the ability to differentiate into lots of different cell types. So stem cell biologists, regenerative medicine experts, all these individuals are now not just using embryonic stem cell lines, but are using induced pluripotent stem cells that were created by adults. So when I was at Stanford, I was at the Stem Cell Institute, so I did a lot of stem cell related research. Still doing stem cells here and focusing on mammalian stem cells taken from fat. We're trying to turn it into different types of cells. But these specific movies are taken by one of my friends that was there. She did herself, went through the biopsy, gave her skin sample, thought, well, if I'm going to be asking subjects to donate and do this and consent for research, I'm going to do it myself and just see what the process is. So she did that, did it to her own cells, converted them into induced pluripotent stem cells. So she had a bunch of those cells growing, and it was a specific line. It was Dr. So-Young McElroy, and so she actually converted those iPS cells into heart-like cells. And you can do this, it happens fairly easily, with a high differentiation rate. So they will cluster up, divide, find each other, start to take on the morphology and physiology of cardiac cells. Okay, cardiac muscle cells and then cardiac conducting cells. That means some that can send action potentials and keep the electrical sort of circuit going, and then some that can actually contract. So they actually shorten. Hopefully this plays the right way. Can you guys see it there? No. Okay, display settings, I'll just change this. Sometimes if I change the view, it, why is it not playing? Okay. Let's see. Second screen only, extend. I'll try one more thing. If not, I can just open it up right from the folder, but I have them linked inside the file, so it should be able to play. Screen only, duplicate. I'm hearing something, it's so weird, because they do have a little bit of audio. All right, that's fine. I'm just gonna pull up the movie in my specific folder. I wanna be able to show you because it's really cool. Courses, and then I have the grab animations. Oh my gosh, it's so hard to do screen. So I had them inserted there. I don't know why I didn't want to play. All right, so now you can see it huge. Those are actually from skin stem cells that were converted into cardiomyocytes. They actually make little colonies, they make little clusters, they make little embryoid bodies. In this case, they actually beat. These are from skin. I don't know if you realize, seems like sci-fi, right? It doesn't seem like it's real, but they actually take on the anatomy, the morphology of myocardial fibers, and they will actually contract. So we got that video, it was really exciting, because I was like, oh, this is like little mini So-Young hearts. Her name is So-Young. So I was like, mini So-Young hearts in a dish, and there's a bunch of them, and they're just beating. Take them out of the incubator, they start to slow down, but when they're in the incubator, 37 degrees, just like the human body, they beat at the same rate as the human heart. So, kind of crazy, right? So I was like, we gotta videotape this, and she had all kinds of publications, and we've had different stem cell publications on. Here's another example of one, little embryoid body, little cardiomyofibers, actually contracting. So they've got conducting cells in there, and the regular muscle cells in there that shorten. This one's going a little slower, and these videos are not the best. We gotta get better at doing that, but it's just very exciting. So we're like, oh my gosh. You could hear us actually in a couple of videos.
Isn't that wild because it's not in your body, it's like in a petri dish and it's beating
Yeah,
on
it
its
is.
own?
Yes, it is very weird, yes. So, yes, and this is to show that stem cells coming from skin, actually, this revolutionized the field. Because in biology, you guys know, in biology and science, there's always these set sort of dogmas. This goes to this, goes to this. That's just how it is, right? You learn it. It's like DNA to mRNA to protein, the central dogma. It is what it is. A cell starts at the embryo, it differentiates, it goes down a developmental path. It is what it is. This actually went, it could go down a path, and guess what? Scientists can push it back the other way. Now you can use this for regenerative medicine and healing, and Even things now that are being looked at are blindness and deafness and spinal cord injuries and neurodegenerative diseases like Parkinson's disease. We talked about that in lab and lecture. Heart disease. I mean, the list goes on and on. So there's a whole bunch of things that are already cutting edge that are going forward right now involving stem cells and the heart. Okay, so that was a little segue and kind of getting outside of the anatomy of the heart. I feel like I need to mention some of these cool things that are going on. Even though anatomy, we know pretty much a lot about the anatomy, we're still learning stuff about the anatomy and physiology of a lot of these structures of the body. Now I believe she has a faculty position over in Chicago. Okay, conduction systems. So you notice those little balls, those little embryoid-like bodies, those mini hearts, they're beating, right? So they're electrically beating, they're depolarizing. Some of them at least are depolarizing, and then the whole ball itself was sort of contracting. Not all cardiac cells are innervated. That means the heart itself doesn't have innervation to every single cardiomyocyte, okay? So remember skeletal muscle, that's different, because remember skeletal muscle, it's a one-to-one connection. Every single muscle fiber gets innervated at that little synapse by an axon terminal, right? So motor neurons leaving the spinal cord down to the brain, and then they activate the one leaving the spinal cord. That motor neuron, every single skeletal muscle fiber has to have a synapse and get activated to depolarize and then to short, right? What is that contact, that synapse called? On the plate, the neuromuscular junction, yes. Cardiac muscle cells don't need that. Obviously, you saw them in the dish. There's no nerves. There's no body parts, as you said. It's so weird. It's not in the body. It's just in a dish. There are no nerves. There are no nothing. It will beat on its own. Skeletal muscle fibers are not able to do that. So the point is, the heart doesn't need innervation in every single fiber. There's only innervation at key places, okay? So it allows the heart to contract in a rhythmic way with no innervation. It'll keep beating, right? It has a beat. It maintains that cardiac cycle. We call that an inherent rhythmicity. So no matter what, it's gonna continue beating, right? As long as there's oxygen and nutrients, it's gonna fire, it's gonna beat. So it's the basis of the rhythmic heartbeat or the cardiac cycle. We'll learn more about in physiology. The heart can beat on its own. Matter of fact, we can take out certain pieces of the heart, or we can take out the whole heart. It will continue to beat as long as it has the nutrients and materials it needs. And this is how, that tissue can stay alive as long as it's kept well. We can do heart transplants, right? You can cut out the vessels, you can cut it out, put it on ice, right? Slow things down and make sure it's cold, transport it, put it in the new individual, attach it to the major vessels, boom, there you go. Then you just gave somebody a new heart. The heart can beat on its own. It doesn't need innervation. So the conduction system is how it's able to do that. So there in the heart, there is conducting cells, and there are myocardial fibers, or some people call these cardio, myo just means muscle, right? Fibers. These are the ones that contract, and they operate the sliding filament mechanism, and they have sarcomeres, and they look just like, a lot like skeletal muscle. These are the ones that are weird. I'm just going to say they're weird, and they actually fire action potentials. They fire and spread, actually, because they're all connected to each other, those action potentials. And they'll do this on their own. You don't need your brain and your nervous system to say, hey, conducting cells, keep conducting. They'll just keep conducting. Okay, they'll keep firing action potentials. And that's what we saw in those movies, those Sun Yun's mini hearts. There are some cells in there that are conducting cells. And they're firing action potentials and sending that wave to adjacent neighbors, and then that connection goes to the muscle, the cardiomyofibers, myocytes. I left out that one. A lot of people will call these exactly what they are. Cardiomyocytes, heart muscle cells. Cardiomyocytes is sort of another term. Those are just the muscular contracting ones. Now those receive, they get an action potential as well because they get positive charge that comes in, calcium floods in, more calcium comes from the SR, and then they contract. But the ones that generate the action potentials are the conducting cells, okay? So they signal all the cells of the heart to contract. The conducting system is actually a series of specialized cardiac muscle fibers, and we call them like conducting cells or electrically active cells of the heart. And they're in specific locations inside of the heart. The sinoatrial node or the SA node, this is the one that sets the inherent rate of contraction. This is the one that generates the impulses. You guys have all heard, I'm sure, at some point of the pacemaker. Have you heard of the pacemaker of the heart? And you've heard of probably electrical, clinically created pacemakers that can be put into somebody's heart, right? That has a stimulator, it's got an electrical stimulator, and they put it right in the right position so it can actually stimulate the heart. It acts as sort of a substitute pacemaker, but this one is the real body's pacemaker. The SA node generates those impulses in the heart to keep the heart beating. Get rid of that. Okay, so if we look at this, and actually, yeah, so in your slides, you might have just a blank page. There's a little blank sheet over it. But if you open the PowerPoint, you'll be able to see this. So the SA node is right up in here. So what are we looking at? What's this chamber? Right atrium. So in the upper lateral wall of the right chamber, we have the SA node. That is the pacemaker, okay? And then we have another node. It's called the AV node, or the atrioventricular node. And it's right at the base, connecting sort of the atrium and the ventricles together, right at that base, that platform. It's called the AV node, okay? So this is another set of electrically conducting cells. Those actually give rise to the bundle of His. This component right here is called the bundle of His, named after, I believe, the anatomist. Bundle of His, and then this splits into the left and right bundle branches. These are called the bundle branches. That's called the bundle of His. And then we have the AV node, and back at the top we have the SA node, okay? So it goes down into the right and the left bundle branches. These go in the interventricular septum, so they're in that wall, and they continue in a series all the way down. All these branching fibers turn into Purkinje fibers that go up into the myocardium in the wall, even up into the papillary muscles, all into the body of the wall of the ventricles, okay? And those Purkinje fibers are sort of the last terminal fibers in this conducting system, okay? So this sets the rhythm, it fires. These are sending action potentials. And you don't have to know the details of how it's done. For now, it kind of seems like magic in physio. We'll go over exactly what happens inside those cells and how they set that rhythm. All you have to know for anatomy is the overall conduction system, how it's structured. It starts here, the firing is happening here at a given rate. It spreads, those action potentials spread through the myocardial cells or the cardiomyocytes throughout both atria. When they reach the AV node, that actually, the impulses sort of pause here. So the AV node has a slower sort of natural rate. It pauses here, and then it sends the conduction of action potentials down the bundle of His, bundle branches, and Purkinje fibers up. So what this allows the heart to actually do is beat in a coordinated way, all sort of in synchrony. So the action potentials generate here, spread, internodal fiber get here, they pause for a second. So that allows the atria, the cardiomyocytes, the regular muscle fibers, to get that stimulus, right? Positive charge comes in them, sodium, calcium, then they're able to contract. So this allows the wave to spread through the atria, and then the atria contract. Pauses for a second at AV node, then travels down the bundle of His. Once it goes up to the bottom of the ventricles and up, so it goes straight down and then up the sides, it allows the ventricles then to contract after the atria have now relaxed. So atria contracts, relax, impulse spreads down, zips up, and then ventricles contract. And actually the ventricles don't contract like from the top down, they actually contract from the bottom up. So this impulse goes, and once it reaches all those cardiomyocytes, the ventricles contract almost like a bellowing or squeezing sort of motion. They go like that. So it goes sort of like that, okay? And the reason it happens that way is so that the heart can effectively get the blood out to each next set of chambers or vessels in a coordinated way. So if you have a crippled conduction system, and sometimes this happens, and sometimes it results in sudden cardiac death or in a heart attack, myocardial infarction, if there's a problem with the conduction system, you can see that you won't get those cardio fibers to receive the action potential and contract. That's a problem. If the heart is not contracting in synchrony the right way, that can result in fibrillation. Instead of coordinated beating, it kind of is, there's contractions going on and it's not a synchronized way. That can lead to a very serious heart attack and even death. So the conduction system is what keeps everything going in those nice, smooth, coordinated ways. Okay, the heart is innervated though. So note that it's got autorhythmicity, it can beat on its own, okay? But does your heart rate always need to stay at the same pace? Like when would your heart rate need to speed up? Exercise or anything physically demanding. Emergencies, remember the 4 F's we talked about? Okay, those 4 F's, I'll come back to the heart. The sympathetic nervous system and engaging that part of the peripheral nervous system actually causes, through nerves going down, so here's the sympathetic chain ganglia, remember the sympathetic chain ganglia with the nerves that leave, the neurons that leave the spinal cord and then go out to different ganglia, go on the second set of nerves, the postganglionic cell. In that case here, they go, sympathetic nerves leave the sympathetic chain ganglia and they go out to the heart on the AV node, right above where the AV node is, right above where the SA node is, and the body of the left ventricle. This is all left ventricle. It's the biggest part of the heart and the strongest part of the heart. Those are all sympathetic fibers. What does sympathetic fibers release in terms of neurotransmitter? Nope. This is the fight or flight one, right? So sympathetic nervous system.
Epinephrine?
Yes, epinephrine and norepinephrine, yes. So mostly norepinephrine, neurotransmitter of norepinephrine is released. That's adrenaline, right? These are derivatives of adrenaline. So that is, those nerves from the sympathetic nervous system are little communicating branches that release norepinephrine, and in those cases allow the nodes, the SA and the AV node, to pick up their rhythm. Fire faster, okay? So that neurotransmitter activates those cells in a way that makes them speed up. So they pick up the conduction of the heart. Now remember, we also have the parasympathetic nervous system. This is the medulla oblongata. This is the cranial nerve. Remember the vagus nerve? Do you remember what number cranial nerve it is? If you do the mnemonic real quick, 1 through 6, 7 through 12. Close. 10. Wait. No, you might be right. Sorry, hold on.
Wait, 8. I think it's 8.
I think you're right. I think it's 8. No, it's not 8. It's not 8. Vestibulocochlear is 8. That's why. It's cranial nerve 10. It should be.
Yeah.
We got to double check this. And I know this answer. I'm mixing it up. Okay, 10. Good. All right. So, gosh, like I'm forgetting the cranial nerve. Cranial nerve 10, vagus nerve. This is part of the parasympathetic, bless you, response. And this leaves from the nuclei in the medulla, goes down, and through this nerve, the vagus nerve, remember this is the unique one that wanders down to the pleura, the heart, the diaphragm, a lot of the GI tract organs, the reproductive and urinary organs. This one has branches that goes right again to the SA node, the pacemaker, and the AV node. It doesn't go to the body or the ventricle. This parasympathetic nervous system is what? What is it responsible for doing? Rest and digest, right? So if you are calm and you're good, the baseline level or low level of where heart rate needs to be will be sort of activated by the parasympathetic nervous system, slowing the rhythm of the heart down, right? So 60 beats per minute, 55 beats per minute, 65. If you're runners or athletes, typically if you have better cardiovascular fitness, you have a slower heart rate. People are always surprised to know that. You can tell more about someone's health and cardiovascular fitness by their resting heart rate pretty much than you can by their activated exercise rate or their maximal heart rate. That's one factor that determines that. So The sympathetic nervous system goes to the SA, AV, and body of the left ventricle, and the vagus nerve, parasympathetic nervous system, goes right to the SA and AV node. And that acts to either slow down or speed up and increase the contraction of the heart rate, of the actual heartbeat. Okay, so blood flowing through the heart. So now we're going to kind of trace the flow of blood through the heart and how it actually works. So beginning with the oxygen poor blood in the superior and inferior vena cava. So remember, the inferior and superior vena cava, they dump into the right side of the heart, the right atrium, right? So they go into the right atrium, it goes into the heart, and then it goes through the pulmonary and systemic circuits. Blood passes through all the structures sequentially, so it goes from the atria to the ventricles, and then it goes out the great vessels or the great arteries. The atria then contract together, the ventricles then contract together. So if we trace the flow of blood through the heart, I like this diagram because it kind of goes exactly, it traces from the vessels, bringing the low oxygen or oxygen poor blood back to the heart, and then how it actually goes through the heart, how it goes to the pulmonary circuit, comes back to the heart, and then how the heart gets it out to the rest of the body in the systemic circuit. So if we trace it here, it's superior and inferior vena cava, dumps into the right atrium. From the right atrium, it fills up, goes into the right ventricle, through the tricuspid valve, remember the three cusp valve, through the tricuspid valve, and then it goes out the pulmonary trunk. That splits into the pulmonary, left pulmonary artery and right pulmonary artery. This is tucked underneath the aorta, the aortic arch. And then that pulmonary arteries, those bring the blood to the lungs, where they go through a whole series of capillaries, tons of capillary beds that are surrounding the air sac. So in the lungs, there's lots of tiny little alveoli. They're made of simple squamous epithelium, right? They're everywhere. And actually the capillaries are surrounding and that are right next to those, so that diffusion can happen readily. So then it comes back, gets oxygenated, picks up blood, comes back to the heart. Do you remember the vessels that return the oxygenated blood to the heart from the lungs? Okay, remember the little definitions? We went over the basic definitions of what's artery, what's a vein. Remember? Pulmonary veins. That's right. Good, Calissa. Pulmonary veins. Pulmonary veins are paired. There's a superior and inferior on both sides, both on the left side and the right side, and they're bringing oxygenated blood from the lungs back to the heart, and it's entering the, what chamber is that? Left atrium. Fills up in the left atrium. From the left atrium, pushes down through the mitral valve or the bicuspid valve, and then fills up into the left ventricle. From the left ventricle, it then gets, with contraction and conduction going through the heart, it gets pushed up and out through this major vessel, the biggest vessel of the body, the aorta, and it goes through that aortic semilunar valve. So it pushes through, goes out the aorta. From there, it goes to the entire head and neck, and it goes out to the rest of the body, out to the systemic circuit. So serving the systemic circuit in the circulatory system. Yes? So I like this because it kind of goes through each segment of where blood is coming into the heart, how it goes through the heart, and then it goes out. And this is making it less complicated, I think, than it really is. Because all this is actually happening at the same time. The atria are filling, they're going down to the ventricles, and the ventricles are pumping and squeezing them out the vessels. So everything is in synchrony. This breaks it down, and we're just dropping one, we're tracing like one drop of blood, right? Because really it's a pump. If the atria fill up together, they squeeze, they send it down to the ventricles get the conduction that spreads up, they squeeze, it goes out the major vessels. So it's going to pulmonary circuit and systemic circuit at the same time, right? This just breaks it down and shows you what is that path that it actually takes. So the cardiac cycle, we'll spend a lot more time in physiology than we do anatomy, but for now, I'll briefly trace sort of how the blood goes through. Cardiac cycle just means the series of events that allow the heart to beat. So I'm going through one beat. Atria contracting, ventricles contracting. That's the cardiac cycle. Okay, so it's kind of the conduction and flow of blood that's happening during the cardiac cycle. So superior vena cava and pulmonary veins are dumping into the atria, right? So blood is filling up into the atria. It reaches a point where blood is filling and it's full, and it pushes on those valves and it opens them, okay? And then it starts to fill into the ventricles. So when pressure is great, builds up in here, opens the valves, blood can then pool into the ventricles. So it fills up, atria contract, helps to shove the blood all the way down into the ventricles. Pressure fills up in the ventricles. These valves, the AV valves, are shut because the pressure is high in there. So they're pushing up against the cusps of the valve. They're keeping it closed. That's good. We don't want backflow from ventricles back up to atria. That's bad. And when you have valve problems, prolapse, where they're going the wrong way or opening too far, or stenosis, they're stiff, you have problems with flow of blood and backflow of blood. That's real blood. We need this blood to maintain either oxygen and no oxygen, right? So it has to be separated. So they fill up, pressure rises. Once pressure is great enough, it actually pushes on the semilunar valves, the pulmonary semilunar valve and the aortic semilunar valve. Those two kind of inside the core valves. Those get pushed open, and so now blood can actually go out through the major arteries, through the aorta, through the pulmonary trunk, and then it goes up here and pressure builds now, and there's a lot of it in there, and it falls back down just a tiny bit. Pressure builds and causes the semilunars to snap shut. This prevents backflow, again. And this is all happening while the ventricles are contracting. Pressure rose in there, snap those shut, opens the semilunars because pressure is great, and with the squeezing or contracting the ventricles from the bottom up, surges the blood out the great arteries, pressure builds, boom, closes the semilunars. This allows the blood to continue forward through the vessels, okay? And this is just kind of enlarging. They're open, enough goes through, and there's pressure building in there, and it kind of falls back down and closes those semilunar valves. It looked like there was blood spattered all over the screen. I was like, what is that? That is a messy diagram. Okay, so the heart sounds in this process, we call this, we can measure these. There's auscultatory positions on the chest, basically the sternum and the left underside of the ribs, upper and underside of the ribs and middle of the sternum, where you can pick up the sounds of the cardiac cycle. And the sounds of the cardiac cycle, real simply, we like to say they're like a lub dub. And it sounds weird, but I had a physiology professor at HSU, I'll never forget, he used to do this. So he used to say, you know, the sounds of the heartbeat, the lub dub, the lub dub, the lub dub. And I never thought the heart sounded like a lub dub, but he's kind of right. And a lot of people describe it as that. It has sort of that lub dub, that sort of sound. When you listen, auscultatory sites across the chest, you can hear it. So with stethoscopes, we're going to do all this in physiology. The first sound is the lub, the second sound is the dub. The first sound comes from the AV valves whoosh, snapping shut. So when you're actually listening to the cardiac cycle through the chest to get someone's heart rate and information, sometimes you can hear valve problems, right? Even with the stethoscope, by listening to these sort of auscultatory sounds that you can hear through the chest. The AV valves are snapping shut. So atria filled up, and then pushes through the AV valves, pressure builds up, and then they snap shut. That's the lub sound. Second sort of sound of the heartbeat is when pressure is building now in the ventricles